One of our board members, and he also leads creation tours, as you just saw a minute ago. But the topic, real chemistry, Ron is the person to speak on that. He is a chemist himself. That's his degree in chemistry. And he's also studied microbiology at OSU, Oregon State University, and has worked in the aerospace industry for way too many years. And he's glad to change his career to creation ministry. And so he's going to speak on this topic, origin of life by chemical evolution. Is that even possible? Ron?
Thank you, Heinz. We'll wait till the slide comes up. I went to school at the University of Washington, but I started my experience in chemistry actually in high school, working before class in the lab at work or at school as a junior, and it just translated into a love for chemistry. So a lot of people, I say, you got a degree in chemistry. That's kind of, why did you do that? Well, it's really an exciting field, but a lot of people don't like it because it's kind of scary. Well, it actually was pretty scary for me too, but tonight we're going to be talking about real chemistry doesn't support the origin of life by chemical evolution. In school, in college, I learned a mantra about chemistry that kind of summarizes what I think about chemistry: better living through chemistry. A lot of what everything we see is based on chemistry and the physics that goes along with it. So I'm kind of excited about looking at chemistry because I see it everywhere I look. And of course, that's one of the issues that is an issue, a significant issue in the evolution theory, because that's, in order to have evolution, you have to have life to start with. So that's what we're going to talk about tonight. There are many issues related to evolution that are actually present itself as an Achilles heel of evolution theory itself. We've talked a lot about those in many of the other presentations we have, like Big Bang cosmology and fossils and genetics and geology, but tonight we're going to focus on the origin of life. Where did life come from in the first place? And we're going to be looking into some of the chemistry behind it to show that indeed chemical facts and processes that are the basis of actual chemistry really don't support the evolutionary theory. It has definitely got an Achilles heel in it. Science, just a general review of this, science is great at, let's see, I got to catch up with this. Excuse me. Science is great at finding out about how things work, but it's not very good at finding out how it started. Basically because science is a real-time technology and field of study that looks at what we see today, but looking back and extending in the past is very difficult, just like other aspects of evolution. But when we look at the evolutionary theory of the origin of life related to chemistry, there really isn't any solid evolutionary theory. But it's important for us that are kind of novices in chemistry to understand a little bit of what chemistry is. Most people are because they're kind of afraid of chemistry because of the technology. I wanted to go through some real basic things. First of all, is just a definition of what chemistry is. Chemistry is basically, in my mind, from a simple point of view, breaks down into four basic sections. One is composition. What are things made of? And the second thing is structure. How are those things that make up the atoms and and electrons and molecules, how is it put together? And then thirdly, what are the characteristics, what are the characteristics that they have? And then the fourth thing, and it's what's really important for life and everything we deal with in chemistry, is how does it behave with other substances or environments that the chemicals find themselves in? So what is the relationship between life, cellular structure, and living systems and chemistry? Well, we know that, and we'll talk about this in just a little while, that chemistry involves atoms and molecules and their reactions and relationships with each other. There's a gentleman named A.E. Wilder Smith who has got more degrees than I want to name here, a lot of PhDs, and he was a chemist in Switzerland, and he had a couple of cool little statements that I think that really summarize kind of the focus of where we're going to start tonight. The pure chemistry of a cell is not enough to explain the workings of the cell, although the workings of the cell is chemistry or are chemical in nature. And that's a real interesting dilemma, because chemistry is actually how life works, but it doesn't actually consist of chemistry. The chemical workings of the cell are controlled by information that does not actually reside in the atoms and molecules. And we're going to talk about a little bit more than that. The model of evolution states that, or basically can be summarized in terms of the origin of life, in that the Earth formed, about 4.6 billion years ago. Chemicals form molecules in a primordial soup or the ocean, thermal vents, or a number of other theoretical places. And then the molecules bonded together to form and make living cells. They were formed by chance over time, and what I call necessity, that is natural laws of chemical interaction. Even though and we're going to talk about this, there's actually no directional definition in chemistry. They just react the way they do. And out of this, by chance over a long period of time, life formed. There's another theory by a comic, you've all familiar with Garfield. I thought this was an interesting little comic that kind of describes the dilemma that we might face. Garfield's History of Cats, the very first cat crawled out of the ocean or the sea about 10 million years ago. And fortunately for him, it was only about another 15 minutes before the first mouse crawled out. A professor named Frank Harold made some interesting points, and I'd like to read these to illustrate kind of the current status of how scientists view the evolutionary viewpoint. Life arose here on Earth from inanimate matter by some kind of evolutionary process about 4 billion years ago. This is a very interesting statement. This is not a statement of demonstrable fact, but an assumption almost universally shared by specialists as well as scientists in general. It is not supported by any direct information. Isn't that an interesting comment? Nor is it likely to be, but it is consistent with what evidence we have, what we do have. And so we're going to be looking at some of the evidence, chemical evidence of how chemistry works and how it relates to, well, how did life form to start with? However, the creation viewpoint is very straightforward. One of my favorite verses in Colossians is in Colossians 1:16. God was very clear in how he presented us. For by him all things were created that are in heaven and that are on earth, visible and invisible. All things were created by him and for him. God created life, and that's the simple creation viewpoint. And we'll talk about that a little bit more later on. I think when we look at the evolutionary model, there's some very key things that really are a challenge. The critical nature of the origin of life in the evolutionary model. In answer to challenges to the evolutionary model, many just avoid the subject. I remember talking to a evolutionist on one of my field trips to eastern Washington, to the Scablands, and we were talking about change over time in species. And I asked him, what about the origin of life itself? And he made a very interesting comment. He said, I'm not sure that it's that important, and we need to, I just need to kind of skip that. And what we really need to do is talk about how life changed once life got here. Basically, what he was saying is that he had no answer for how that happened chemically, how life started. Some argue, and that what he was doing is what the second bullet line says, some argue that the origin of life is not part of the theory of evolution. And I don't think either response is very logical, but it's understandable considering the evolutionary origins are best inconsistent with and actually contradict real science, especially real chemical science. My opinion is, and I think it's very important to consider this and remember this, the origin of life is an absolute necessary precursor for all evolutionary model discussions. It has to be, because of course you can't have changes in species if you don't have species to start with. Where did life come from? So the purpose of this presentation is to discuss and examine the origin of life and illustrate that evolution is simply without scientific foundation. The origin of life remains a mystery to the evolutionary scientists. And there are myriad of scientific theories, but nobody agrees on which one is the valid one, mainly because they all contradict chemistry. And we're going to talk about that. Biochemists have been able to show even how the simplest cell could have been formed without intelligent cause. Before we get into some of the problems with the evolutionary theory from a chemical point of view, I'd like to go through some just basic terminology. And this is a little bit complicated. Let's see if I can get the right slide up there. But I want to go through some of this. It's going to be a little bit complex, but I want to make sure you understand that you don't have to remember a lot of this stuff. And part of the purpose is to show you how complex it is. And these are just basic information for discussion purposes later. Atoms are the basic unit of matter, you know, like carbon and hydrogen and oxygen. Molecules are the specific arrangement of those atoms, you know, like water. It's very straightforward and simple. Amino acids are very key in life systems because they form the foundation of proteins. And that is a picture of what an amino acid is. It's on the left-hand side is a nitrogen group, and the other side is a carboxyl group, and that's the acid part. So that's why you have an amino acid. And then in life systems, there are 20 of them that are used, and the difference between them is that little R group down there at the bottom in the middle, what's called side chain. Proteins are made of, like we all know, chains of amino acids that are bonded together linearly. This is a rather complex picture coming up here, and this is DNA, deoxyribonucleic acid. And of course, this is the blueprint containing the genetic information. Most everybody knows that. Looking at this, wanted to just show you the complex structure of it. And remember, you don't have to remember this, but just kind of keep it in your mind that generally what we're looking at. On the left-hand side, where the three arrows are showing, those are pointing to the backbone, which is a deoxyribose molecule with a phosphate group. And then the pink and the green and the dark blue represent the nitrogen bases that actually form the code of the DNA that are hooked to the backbone. And then on the right-hand side, you have another chain, and that's what DNA is two chains hooked together, actually in a helix, and it's a kind of a duplicate, if you will, of the other side. And they're bonded together with what are called hydrogen bonds. What's interesting to me is that in the DNA, there's roughly about 3 billion base pairs. Can you imagine that, 3 billion? And stretched out, it's about 6 feet long. If you take it out, take all of the DNA in your cell and stretch it out, it's about 6 feet long. That's a lot. Here's a picture on the next slide of down on the bottom of that DNA, and part of it has been untwisted and expanded. And each triplet of the bases form what are called codons. And each one of those codons, the 3-letter bases, are specific for each amino acid that are used to make proteins. Are you with me so far about that? So when the cell reproduces, it untwists, the DNA untwists, and another protein comes along and reads that code and spits out information that goes outside the nucleus for each one of those specific amino acids in order to be bonded together to make a protein. And each one of those sections of a group of codons for a specific protein is called a gene. So you have the codon, you have the DNA, the codon, the gene, and then you've heard of chromosomes. Those are just groups of DNA pieces that make up all the DNA of the cell. And that's a picture of the chromosome up there at the top, where the DNAs are grouped together in groups and bundled in a cell. It's amazing to me how complex it is. And up in the right corner, I'm showing you some of the samples of the codes, or the codons, that represent the various amino acids. For example, UGC is for cytosine, and CUG is for leucine. Those are the names of the different amino acids in the sequence of proteins. So it's rather complex, isn't it? But it's interesting, it's all based on chemicals. And it has all this information laying on top of it. Again, we're thinking about where did that information come from to start with? What's the origin of that information? And what's the origin of the systems that put it together and it's read? The information is translated into what's called a messenger RNA, which is called ribonucleic acid. It's a relative of DNA. We don't want the DNA outside the nucleus in the cell where it can be damaged. So a copy of it is made, kind of like a mirror image of it, and sent outside the cell into the factories where the proteins, and that's the messenger RNA's job. The A and the G and the U and the C are corresponding code with the similar bases to the DNA code. And that goes outside the cell. That messenger RNA is that little line on the bottom here. And you can see one of the codons down there, GCC. And what's called a transfer RNA is the little chemical chunk that carries each different amino acid. And it's floating around in the cell, and when the machine reads the particular codon in the messenger RNA, then it grabs the appropriate amino acid in this complex molecule called a transfer RNA. It's kind of like little trucks carrying the amino acids around in the cell. That's what the tRNA is. And it gets a little bit more complicated because then that's done, that's all meets together. The transfer RNA, the messenger RNA meets together in the little structure, not a little structure, but a very complex structure in the cell called a ribosome. And that's where this business of creating the protein is produced. And here's a picture of it. The little pink guy is the ribosome, and the messenger RNA kind of is glued to it or stuck with it, and then the transfer RNAs bring in the amino acids as the messenger RNA is read, and the protein is produced. Let's see. Go back to the right one. So that's pretty complex. Everybody understand all that? You got it? Well, don't worry about it being so complex because you've probably forgotten most of what you just saw. But that's okay. The idea, one of the ideas is to show how the basic simple structures, if you put it in a presentation like that, are not that simple, are they? There's a lot involved behind that. So now we're going to look at some of the problems that I think, present kind of the facts of the Achilles heel of the evolutionary concept from a chemical point of view. But first we're going to talk about something I think is probably one of the most important things, and that's related to the information problem. Where did the information come from that prescribes what happens in the cell from an origins point of view and also from the function that we see today in our life processes? Information is everywhere, isn't it? Does everybody understand clearly what information is? Well, we're going to talk about that. The question is does chemical evolution, time plus chance, align itself with our knowledge of information science? Remember that evolution is based on time plus chance, isn't it? Right? So what is chance? Well, chance actually is not a thing. It's really not an entity, and that's something important to remember. It's not an active force. It's not a driver. It's not a being, if you will. It's not an agent of causation. You got to remember, though, that that's the fundamental process in the evolutionary concept is time plus chance. Actually, chance is really the possibility of something happening, or the word is probability of something happening. It's an accidental event, actually. A chance occurrence is to do something by accident without design or without design. It's an outcome due to outside forces, actions, or laws, or entities. If you roll dice and you have a chance of getting, let's say, a 3 and a 6, like it says there shows on the slide, what causes that to happen? Maybe it's how you throw it. Maybe it's the wind in the air, the weight of the dice, the angles you throw it at. But it's not inherent in the dice itself. So what is information? Is order, data, and organization really information? Well, I think not really. You look at a crystal of salt. Is there order there and structure? Yes, that's true. How about a protein sequence? There looks like there's organization there, there's order, and there's some data there. But is that information? How about an ice crystal or a snow crystal? It looks pretty ordered, and there's a lot of information there. But those are all just determined by physical constraints. There's nothing inherent in the particular properties of the material or the material itself that cause those things to happen. Or is that true? It is actually the chemical properties that do that, that drive this. But it's not information. It's just order and data. What is information? I'm a pretty simple guy, so I found this picture and I thought it really communicated to me what information really is. It's a code, it's a language, it's knowledge or alphabet that's sent, received, that's understandable or executable. We have a sender and a receiver. Excuse me, this back up this slide. This is just order and data. If you look back at these items here, but there's no meaning to those things particularly. So, well, if you think about what information is, it has to be useful. There's an ability to read it and then interpret it and then execute it. It has to be functional and prescriptive to be real information. A salt crystal has data and order, but it really doesn't communicate anything. It has no real useful information in it. If we look at that picture right there, does everybody recognize what that is? Is there information? Is there data there? Is there order? Yeah, we have 3 different colored lights, kind of a structure to the piece of equipment. But everybody knows what that means, don't you? What does the red light mean? It means stop, doesn't it? The yellow means drive fast before it turns red, right? The green says you're okay to go. Everybody knows that system. There's order there, there's data there, but it's also true information because there's the piece of equipment that's the data actually is sending a message to the receiver, right? Us. Because we know what the laws are regarding those, and we know what those signs mean, what those colors mean to us. So there's a purpose for it, and it has a useful and practical and instructional and functional purpose, and we know that, as compared to just a salt crystal or even a protein molecule. That's just data and order. Useful information in a cell. The cell is full of prescriptive information. The DNA and RNA and proteins and other molecules execute cellular action with meaningful and functional output. Like, you can see how complex the cell is in that little picture, and obviously it's much more complex in terms of the chemistry. So it's good for, there's transportation that's done, structural work, metabolism, enzyme action, repair, replication, waste management, cellular excretion, and intracellular communication. And it's all done chemically. Okay? So that's pretty complex. It has useful information. But where's the software? What's the software behind it? Like A. Wilder Smith said in the original slide, or one of the first slides, atoms and molecules do not contain an integrated information system. They're just dumb stuff. That's what I really like to call it. I mean, it's complex, and it does interact with each other in the environment, but it really has no direction in and of itself. So if that's the case, how did we get from dumb stuff to an integrated cellular system like we see here? Because that's all chemistry. It's all atoms and molecules. So where did the information come from? Well, Mr. Wilder Smith is going to teach us a new word today. It's called teleonomy. He says that teleonomy rides on chemicals and chemical process, but it does not arise from the chemicals themselves or cellular information. That's kind of a word that's derived from 2 Greek words called It means end purpose and law. It means end-directed or purpose-driven law. It's the active operation program or force in living systems. He says that riding on top of the atoms and the molecules which react each other in natural necessity, but they don't do it with any particular direction or end purpose. However, he says that riding on top of those is what's called teleonomy, this purpose-driven force. It's not physical, but it's metaphysical, because again, it does not ride on the chemistry. It's programmatic and computational aspects of purpose that ride on the chemicals. The quality of purposeness and a goal-directedness of the structures themselves. Here's a picture on the top. That's an example of just a really nice living forest like we'd see here in the Northwest on a lake. But if you look down below, what's the problem with the picture below? It's the same forest, same chemicals, same sunshine, same information in the structure, and the atoms are the same, but there's a problem. The forest is dead. It's not functioning. Something's wrong. But you got to remember the same stuff and the same information is present in both. The issue is the teleonomy is missing. The force, the driving purposefulness that rides on top of the chemistry. God made the world, and we see the world as a cause and effect world. Chemicals and information are really not enough. If you wanted to raise, for example, and here's an example I came from a farming background. If you wanted to raise corn as a business with a high quality yield, what would you need? Well, the cause, because we want the result, don't we? Well, first you need the stuff, don't you? You need land and seed and equipment and fertilizer and water and sun. But that's not enough. We need the knowledge that goes along with it. And we actually need a skilled farmer to execute the plan. You can't have just one and two. Think of those as the chemicals. The farmer is outside the stuff needed and the knowledge required. He uses the stuff to activate and implement the plan. He's kind of like the force, right? Otherwise, you can have all the stuff you want in 1 and 2, but nothing's going to happen. The stuff that we're talking about in terms of life are the atoms and the molecules and the laws and the energy. The information resides in the code, but that actually needs something else. It needs a skilled source from the outside, just like a farmer. The teleonomy or the force. Who is that force or that skilled source? Remember Star Wars? The force be with you, right? That was the mantra. You remember when the planet Alderaan was destroyed by the force, by the Death Star? Obi-Wan felt a disturbance in the force. Do you know what the force is actually? In the first Star Wars, they told us what the force is. Every living system is full of what are called, I think it's called metachlorians. They reside in each individual life system, each individual. And that's the actual driving force that keeps all life living. It's interesting that they understood that the life force does not reside in the stuff. So what's the real source of life? And again, this is the major serious problem for thinking about how life started to begin with. Because information and stuff is not enough. Because it doesn't reside in the chemicals. The real source of life. Then the Lord formed man from the dust of the earth and breathed into his nostrils the breath of life. As you do not know how the Spirit comes to the bones in the womb, so you do not know the works of God who makes everything. He is before all things, and in him all things hold together, upholding the universe by his word of power. The effect on the evolutionary model, there are serious issues how matter and energy arose out of nothing. The prescriptive information did not originate or was not generated by the chemicals and processes to start with. And that's all that the evolutionary model has. It has the stuff and the laws, but it doesn't have the driving purposefulness. The skilled source is required to initiate and maintain life processes that does not reside in either the stuff of life or the prescriptive blueprint. And that's the key thing, the key problem. How does this affect the evolutionary model of the origin of life? Well, where did it come from? If it was just left to random molecules. They're not that smart. Perhaps the model of evolution should be modified or perhaps even abandoned. That was kind of the longest part of this presentation because it's probably one of the more important critical things to remember is that the purposefulness, the goalness, the source does not ride in the chemicals themselves or the processes. Although those are natural, quote unquote, they don't yield or get to where we want to get. But we see life operating on those things. And it's embedded in the chemistry, but it's not part of the chemistry. Thermodynamics is probably a big problem too. Everything goes from complexity to disorder in our world. So if you have inanimate chemicals that are just there doing their thing, in order to grow in complexity, they need basically four things: an open system like the earth, an energy source like the sun. I better catch up here. Is this working? It looks like it's frozen. Excuse us for a second. A little technical difficulty. Shall I scroll through it? Sorry, everybody. Quick review. We'll get there in a second here. Okay, there we are. So we need an open system, we need an energy source, number 2. Number 3, we need a way to capture and store the raw energy. And 4, an energy conversion system that takes the energy, the raw energy, and directs it in the right direction. The problem outside the cell, or before cells formed, and before the cellular processes were established, conditions 3 and 4 just didn't exist. There was no mechanism for capturing the raw material energy and converting it in the direction that life requires. This gentleman, Neil Broom, said a fundamental that science has never been able to solve is how to produce energy flow through the system to do this work of coding to produce, for example, functioning protein. Living systems do, of course, harness energy for this purpose. And here's the key, but only because the required purposefully assembled metabolic pathway is already in place and functioning. If you just have a group of chemicals sitting out in the ocean, there's no driving force to use energy in a purposeful way. It's a similar problem to the fact that the information does not ride on the actual atoms and molecules. So it's a significant problem. That was problem number 2. Problem number 3, it does get worse. It's called, we're going to talk about some chemistry here, the hydrolysis problem. Where did life start? Did it start in the ocean? How about a primordial pond? How about pond scum? I've heard all of those. The ocean or primordial pond actually is the worst place in the entire world for life to start due to a process called hydrolysis. Everybody know what hydrolysis is? What's hydro? Water. So it has something to do with water. And here's the hydrolysis problem chemistry-wise. Remember the proteins, the amino acid structure? On the top, you have 2 amino acids, one on the left and one on the right. See the little red box there? When they react together to condense together to form a protein, one of the products is water. That's on the little arrow that goes down in the middle. You see the little water molecule that's kicked out. And you form down at the bottom the start of a protein. Okay? That's called dehydration. Why is it called dehydration? Because in the process of joining those 2 amino acids in the top, where it says peptide bond, you kick out a water molecule. Unfortunately, it's a reversible reaction. The unprotected, and this is key, outside the cell, unprotected proteins are actually broken apart during a hydration process, which is the reverse of that. Dehydration to form a protein, or hydration to break it apart. And it's done in the presence of what? Water. So, if this all happens in the ocean, do you see a potential problem? Or in a pond? So you want to form proteins with a dehydration process in a water environment. This is not a good plan. It sort of is a real problem chemically. Let me see, go back to that. Inside the cell, even though the cell is full of water, it's a protected system. And that's the difference. And that's why it works inside the cell. But it just didn't happen by chance to start with, because it's chemically not functionally proper and doesn't work that way. Another problem, problem 4, and you guys have heard about this, the left and right-handed issue. Look at your hands. You got a left and right hand, and there's no way that you can match them. They're mirror images of each other. Same thing with amino acids that are naturally formed. They have left-handed and right-handed versions of them. They're mirror images of each other. However, in our bodies, in all living systems, only left-handed amino acids are used. If you look at any protein, they're all left-handed. However, naturally, in an open system, without any kind of process to direct it, they're 50-50 left and right-handed, and the problem is that amino acids will react the same no matter which handedness they are. So you will find in nature proteins, if they form, will be mixtures of left and right-handed amino acids in that chain. Well, proteins in an open environment would be made of left-handed and right-handed amino acids. Mixed-handedness would cause proteins not to be biologically active, unable to fold into the right shape to act as enzymes. Cellular proteins are left-handed, and this is something I just recognized a couple months ago. I know exactly why all proteins are made with all left-handed amino acids. Does anybody know? Well, the reason is because DNA and RNA are all right-handed in their structure. That's pretty interesting to me. Because amino acids, you remember, are produced, think back to that process of how amino acids are formed. They're formed by reading the DNA. And that's why they're all left-handed, is because the DNA, individual base units that form the codons, remember, are all right-handed. And I thought that was pretty interesting because it dawned on me why that would be very important when because you remember how the amino acid is read by enzymes, and the messenger RNA is spit out from that. And if you had a mixture of DNA in the DNA of right and left-handed bases, you'd come to a screeching halt. The protein wouldn't know what to attach to and how to read it if they're backwards or mirror images of each other. So they have to be all the same way. But it kind of begs the question, you got a protein that's 150 amino acids long, but you have a DNA that's 3 billion base pairs long. That, to me, is pretty complex to get them all in a right-handed version. How did that happen naturally when they're all in a natural environment? A mixture, 50-50, just like the amino acids are. Not outside the cell. It's just not going to happen. You can't do that because they all react the same. The chemistry is the same. It's just, it doesn't matter whether they're left or right-handed, they'll react the same. Inside the cell, it does work. That's the way it is inside the cell. It's a definite design miracle, in my opinion. And the answer is yes, it does work inside the cell, but not outside the cell. So it begs the question, how did it happen naturally? It really violates a major chemical problem here. This is a kind of a complex problem, or a diagram that you'll see next. And the intention here in number 5 is not to learn or see the system here, but to recognize the complexity of it. And so I'm going to make it very complex. There's an irreducibly complex problem, complexity problem. And we'll talk about what that means. And the example I'm using is called cellular metabolism. Inside each of your cells, you have to take energy from energy foods you eat and convert it to energy that's useful in the cell. So the question is, how does the cell do that? It's a real simple process. So here we're going to go through the process real quickly. Again, I don't want you to know the process because you'll never remember any of this. But just look at the complexity of how it works. It's a 3-step process. The first one's called glycolysis. If you look at the pink, you have glucose right in the middle of the diagram at the top. And you go through a 10-step process. And again, this is the first step. A 10-step process is using 10 enzymes and some ATP molecules, which are energy sources from the cell. And you break the glucose down and end up with a chemical called pyruvate. And that's in the yellow over there. Okay, everybody remember that? There's a test afterwards. You got the paper. This is step 1 in how the cell builds and uses energy from what we eat. The next step, step 2, is called the Krebs cycle. So if you notice up there in the upper left-hand corner, it says glycolysis, and then you have pyruvate. Remember, that's the product of that first step. Now it goes through another 10-step process, kind of in a circular fashion in this diagram, to break that down even further. It uses 10 other enzymes and other reactants. Very complex, isn't it? You guys got this memorized? Okay, again, the test is coming. So you got step 1 is the glycolysis cycle, step 2 is the Krebs cycle. We're not done yet to produce the energy for our cell. Now we have what's called the electron transport chain. That's step 3. This is where what's called ATP is actually made. That's the energy source that the cell can use. You can't use glucose and you can't use starch directly. It has to be broken down. So the next step is actually has 4 parts to it, where the product of the Krebs cycle now runs into this last step. And you end up at the end there with a whole bunch of ATP molecules being produced. Each glucose molecule makes a whole bunch of ATP molecules. But it goes through this complex process. This is what happens in the cell in what's called the mitochondria. That's where all this is done. And it's happening right now for you guys out in the audience. And guess what? How critical is each step in this process? Does anybody know what happens when the spy takes a cyanide capsule? What happens to him? He dies, doesn't he? Do you know why he dies? Well, it happens to be on the last step in that 3rd process. See where the X is? What happens when the cyanide gets into the cell, it disrupts the last enzyme right before the ATP molecules are generated. And guess what happens when that happens? The cell dies. And guess what happens to you? You die. So let me ask you, how critical is each step? It's pretty important. Now think back in the primordial soup, all this happening simultaneously together, all at once, in an unprotected system. It's a real serious problem. And I think this is a very good example of how complex things just don't happen by chance. It's kind of like a roadblock. It's kind of like that big rock sitting in the middle of the street there. It's just not going to happen. All this is a product of random, non-directed chemical reactions in a deadly primordial soup. It's really beyond all scientific reason, from a chemistry point of view especially. Because if you remove one enzyme, one ATP, or one reactant from any of the processes, it's just not going to happen. So they all have to be in place at the same time simultaneously. And that's not going to happen in an open environment without some kind of process that will control it, like it's in the cell. So how did it happen originally? It's a real serious question. There is no answer for that. There is no chemical answer for that. How much time do we have left, Heinz? Okay. The last problem we'll talk about is what I call the chicken or egg process. And we'll end with this one. So, here we have a description of, or a picture of, the double helix DNA, and that's that kind of wound up center line, if you will. And in order for a protein to be built or manufactured, what happens is a large protein called RNA polymerase runs down the line of the DNA in the gene, remember the gene? And it unzips the DNA molecule as it's moving down, reads the DNA code, the codons, generates the messenger RNA, remember that? And the messenger RNA breaks loose and goes out into this cell structure to build the protein. But as it's reading that, it reads down the code, and then it rewinds the DNA. That's pretty complex, isn't it? Now, where did the RNA polymerase come from? It's a protein itself. So somewhere, sometime, something had to read the DNA molecule, because guess where the blueprint for the polymerase is? It's on the DNA molecule. So what read the DNA molecule to make the thing that reads the DNA molecule? Does that make sense? It's kind of a chicken or egg problem in my mind. Which came first? The thing that reads the DNA blueprint, or the DNA blueprint that makes the molecule that reads it. You can't have both at the same time chemically. They have to be there together at the same time. I'll skip the next one. And it's similar with that ATP molecule. Remember the energy molecule? Over on the left-hand side, the glucose metabolism requires ATP to function. But it's the first step in the generation of ATP. So if you need ATP to start the process, you got to have ATP to start with. So which came first, the ATP or the mechanism for creating the ATP? It's a similar problem. And there's some other problems. We'll skip those because of time. I put down 12 of them. Probability, I think, is an important one that we'll just touch on real quick. And that's another serious problem when it comes to life systems. It's been calculated, and you can argue about the numbers, but what are the chances of getting biologically active molecules? Well, a protein, 10 to the 164th power. A DNA sequence, 10 to the 4,000th power. A simple cell, 10 to the 40,000th power. But most people in probability science know that anything over 10 to the 50 is totally impossible. Where does it all lead, all this information, all the problems? I mean, and I left out a lot of them that I came up with, and it's just a never-ending list of them. I thought of a lot of them besides what we talked about. Einstein said, this is a quote actually from Wilder Smith, thinking about Einstein, claiming not to be a Christian, he concluded that the universe, and therefore biology, must have its origin in understanding, thought, concept, mathematics, intelligence, and teleonomy, and not in randomness, chance, plus the inanimate laws of nature. For to him, the greatest miracle of the universe was that we can at least comprehend it. Dawkins was asked by Ben Stein in the interview that he looked at with him, how did life start? And our friend Mr. Dawkins said, well, I don't have any idea how it started. Perhaps it was seeded on Earth by a higher alien intelligence. The aliens did it, so to speak. That's a real issue. What does it all mean? Since any biological change is dependent on life first, originating by natural processes, violating basic chemical laws, the entire model of evolution is without a foundation. I like Mike Riddle, we've all heard from him. He said, why should I accept evolution when you cannot produce evidence? I already have a faith, tell me about your faith, and I will tell you about my faith. There's a logical deduction in my mind. It's rational to believe that God and not unknown events created life. There are 4 takeaways. Origin of life is foundational to the debate. We can't ignore it. Design, intelligence, and information drive life outside of chemistry. It doesn't reside on the chemistry. It comes from the outside. And no evolutionary pathway to life exists. It violates and contradicts chemical and physical and probability and informational laws. And 4th, the chemistry of life confirms God as the creator. There has to be a source outside that's not part of the system itself, because it's not there. And that's our task, and what I would do is to learn a couple of these concepts, chemical processes like hydrolysis and left and right-handedness, and maybe a little bit about information science. And with that, we can be prepared to make a defense when it comes to the origin of life and therefore the foundation of evolution. And it kind of crumbles when you look at it. Mike Riddle said, I like these when he talks about asking questions. How do you know that it's true? Has it ever been observed? Are you making any assumptions? And there's a lot of non-truth, no observation, and a lot of assumptions in the origin of life from an evolution process point of view. So I think it's important that we just learn to think. And I think we'll be finished just how to prepare. You can get involved in learning a lot of these things, but it just takes a little bit of getting a few facts, and that's all we need to really do, and be able to challenge the evolutionary process from all of those concepts. But chemical processes, I think, are one of the easier things to talk about because it's the foundation. So I think we're done.
Thanks a lot, Ron. A lot of information to cover there.
Ton, too much.
And I was looking for some questions online, but we didn't get them.
Okay.
We have a chat window set up. But anyway, you're a chemist, and you talk to other chemists. When you try to discuss this issue with fellow chemists, and they're not believers, how do they respond?
Well, most of them put the question off. Like I was telling you about, the people that I've talked to that are science-based go directly to speciation and genetic changes, mutations, and such like that. And they basically, I really haven't had anybody that's been able to defend it from a chemical point of view, because there's no consistent model in evolution that has been presented that's viable, let's put it that way. And that's the real struggle. You can argue about the different details, like about hydrolysis and environments and the cell structure itself. We're learning a lot. It gets more complicated as we learn. But then translating that into an open system in a pond, it's very difficult to come up with a concept that is anywhere near being viable from a chemical point of view. There's just not an answer for it.
Right.
And that's why it's skipped.
Yeah.
Does that make sense?
In fact, you'll probably get some evolutionists who will say, well, that's not part of evolution.
That's exactly it.
They assume that there is some first life.
Yeah. Interesting, though. Last week I was looking over this, and then I went to a grocery store, and there was a young kid standing there, and he saw my shirt. I had a chemistry shirt that I got for Christmas. It says, oh, chemistry, and it had a tree made of the periodic chart on it. And he looked at me and said, oh, that's really cool. I like chemistry. I took it in high school. He's a young kid, about 20. And I said, very interesting. He was the security guard at the store. And I stopped and talked with him. And he asked, do you like chemistry too? So I explained about my background. And then I asked him a question. I says, what do you think about the origin of life? How did life come about and start with? In a primordial soup or in the ocean. Do you have any thoughts about that? He said, yeah, the proteins were formed in the DNAs and amino acids, and they all worked together and formed a cell over a long period of time. And so then I brought up 3 points. One about hydrolysis, for example. I went through that real briefly with him. And he looked at me and he said, really? I never thought about that. And then I talked about left and right-handedness. Really? I didn't know that. I've never thought about that. And then I talked about information, just real briefly. Where does the information reside? Because we know, are atoms smart? And he said, well, maybe, I don't know. I've never thought about that. And at the end of the short discussion we had for about 5 minutes, he said, I don't know if the evolutionary process, origin of life, really is valid. I've never thought about it before. I've never heard about those things. And it was just 3 little simple points that I brought from the presentation that I remembered that are, because it's so complicated. I mean, there's a lot of weird stuff. That
but there are some very easy arguments.
Very easy arguments.
That you have in your presentation here too. Yeah. And people don't think about those because they take a lesson about Miller's experiment.
Mm-hmm.
Look at it produced amino acids, but they're not told that it produces the wrong amino acids. Produces 50% left and 50% right, and they're not the components of life.
Oh yeah, exactly.
That you will not find that in the school textbook, and that's the problem.
That's true.
They're not asked to think critically.
And even, you can reach into the information part of it or the force behind it in that discussion too, because that particular experiment was very designed for a purpose of leading to where they wanted to go.
Right.
And that does not represent the open system in an ocean,
right,
where there's no smarts there. Atoms and molecules are dumb. They just do what they were designed to do, and they do it really well. I've heard that somewhere, haven't I?
And the experiment produced a lot of tar, useful stuff, and it
Yes.
had to be removed, or else you couldn't continue.
Well, actually, they had to keep it out of the water too.
Yeah.
Did you know that? They had to keep draining it off so it wouldn't go back through the system in the water, because it would have hydrolyzed again.
Right.
So
That was not a random experiment.
no, that was not a random experiment. And the cool thing about the way God has done it is He said, I need a protected system inside the cell to do everything. Mm-hmm. Because all the chemistry that I'm using, those same laws that He created in the open environment, work really well in the open environment. But for life processes to work, they don't work well out in the open environment. The chemistry inside the cell, the processes and the way it's designed, is designed to not only do the job, but to protect each component and step in the process from what naturally would occur out in the environment.
Yeah.
And that's a really key point to understand.
Yeah. And there has to be a membrane to be able to do that. Where did that membrane come from in the cell?
And that's just one thing. That's just one part of it. Yeah.
Right. Yeah.
Yeah, and everything that goes with it.
Yeah, so I think the conclusion is most people just don't think about these issues at all. They just assume.
Yeah, exactly.
They just assume. They don't ask the critical questions.
So if we go with 1 or 2 facts, just simple facts, and ask them, what do you think, and how that fits or how it doesn't fit, and let them draw that conclusion. Yeah. And then we can come to, well, maybe there's, and I told them about the Force, about Star Wars. I think that's a great story about the midichlorians, because they knew that the stuff did not create life, nor did it control it or let it work.
Right.
It had to be something outside of the stuff. Yeah. And so they invented midichlorians. Well, I think God is the midichlorian. He's the one that breathed life into us. And from there, then I can go right to the gospel. It's really simple to do that. Yeah.
Okay, well, thanks for that discussion. It was very useful because if you ask the right questions, people just do not have an answer to those it's really simple questions.
They can be really simple, yeah.
Yeah. Now, you had a few points in the last slides, and if you allow me, I'll upload the complete presentation so people can look at that later on.
We only went through about 6 of the problems that I listed, but there's and those are just 12 that I came up with. I mean, there's 12 more and 12 more past that too. Yeah.
Yeah. Okay. Thank you, Ron.
Okay.
So let me just conclude and just introduce the speaker who we have next month, and Maureen O'Kane. She's presented here before, and she's actually also one of our board members. So she's going to speak on the topic of black holes. Menace or myth? Do they exist, and do they pose a problem? And so just a few questions that she will address, the issues, the role of black holes in space. Do they actually exist? Black holes that challenge our theories, research and comments from scientists, and what does the Bible say about collapsed stars? Those are the questions that you will answer next meeting here, which is on August the 21st. Maureen comes from a different perspective. She claims she never started reading the Bible until she was 25 years old and became convicted and became a believer. And having some interest in science, she started to study science. She is not trained as a scientist, but as a layman, she has asked the questions about science and has come up with a number of good presentations that present things from a layman's perspective rather than from a science perspective. So that's next month. If you're online and you receive the invitation to this meeting by somebody else, you can subscribe to our mailing list online. Just go to the web page and you'll see a subscription there. And also you can always look to see what talks are coming up, which speakers are coming up, and you'll find pointers to a lot of evidence and places to get answers for questions that you may have. So take advantage of that. So let me just close in a word of prayer and we'll be dismissed. Father, we thank you for this time. We thank you for Ron's message. And it's a very critical piece of creation because you are the creator of life. And secular scientists have tried to show how life could have formed randomly with material that you had created, but they have never come up with a solution that is even half close. We just pray that people will use this information to help them witness to their friends and colleagues and family about the message of Jesus Christ and creation. Thank you for this time. In Jesus' name we pray. Amen.